Welcome to Zoominario. Today we're very excited to welcome Gustavo Ballen. Gustavo is a Colombian evolutionary biologist. He earned a bachelor in biology from Universidad Nacional de Colombia and a PhD in systematics, animal taxonomy, and biodiversity from Universidade de São Paulo. His postdoctoral experience includes a position at the Reis group in Queen Mary University of London, and now FAPESP postdoctoral fellowship at the Oliveira group in the Universidade Estadual Paulista, Botucatu. Thank you, Gustavo.
Thank you very much, Claudia, for having me, and thank you all for coming. I will talk about one of the lines of research that I'm most interested in, which is how do we tell the time of macroevolution. But first, I want to introduce this question by asking some things, like how old are these things and how do we know? So for instance, we have here Metallica's S&M album, which was recorded between the 23rd and 22nd of April in 99. And then we have James Hetfield also from Metallica, which we know was born on the 3rd of August, 63. And as far as we know, he's still alive. Then we have the Capriola Notebook from around 1570, housed at the Newberry Library in Chicago. And then we have Vincenzo Capriola himself, that is thought to have born around 1474 in Brescia, and then deceased after 1548 in Florence. This is not actually Capriola, but another Brescian nobleman. And finally, we have the Seiklus epitaph, which is one of the earliest records of musical notation, which is a very short song from the 1st or 2nd century Anno Domini, housed at the National Museum of Denmark, and we don't really know who Seiklus was. Finally, we have this small pectoral spine fragment, whose provenance is unknown, and therefore its age, and we sort of know it might be a catfish, but we don't have any other details. So this shows that as we go back in time, we start having more and more uncertainty on the time of objects or events also. And this is actually what happens with biology also. So why is it useful to know the timing of evolutionary events? First, because they can be used for modeling pandemics, as was done with the SARS-CoV pandemic by some teams in Europe that applied methods using time information for estimating, for instance, important epidemiological parameters. But these methods and timing is key when measuring how fast or slow speciation or extinction goes along the time of the tree of life, in this case, exemplified by this study with lizards. And then we also have time having a key role, or playing a key role in telling us how was the earliest ways of life, was what we call the last unique common ancestor, which is a study from earlier this year that used divergence time estimation to time calibrate the phylogeny and then make inferences on the metabolism of these earliest ways of life. And then where do we look for information on time? We essentially have the fossil record. So we can use, for instance, fossils themselves, but also droppings or the things that the footsteps that animals leave in the sediments, as well as the animals themselves. And all of these remains have some time information from where exactly. One important way of telling the time in the fossil record is using radiometric dating, which is essentially using the relationship between isotopes that undergo transformation one into the other, and then using those proportions as a way of measuring time, because we know these things decay or undergo transformation one into the other in a regular way, and we can measure it. So then we can tell by, for instance, the amount of argon that we find in sediments, if they are old or not, because they tend to be rich in argon as we go back in time and the sediments are trapped. And we can use that information and statistics in order to represent not just the time information for fossils, but also the uncertainty around those estimates. And then we can also use the correlation between the layers of rock in the fossil record in different regions in order to build a general frame or time scale in which we can see different communities in different time intervals, and then tell time relative to other layers in the fossil record. This is, of course, less accurate than using radiometric dating, but it's by far the most common way of telling time. And then, of course, we can use mathematics for modeling the fossil record itself, and then use it for estimating the origination time using the patterns of occurrences in time from the fossil record for a given lineage. Then we can integrate all of these through phylogenies and mathematical models, but how? So this is a phylogenetic tree which depicts the evolutionary relationships between organisms, and it has essentially two very important parts. One of them is terminals, which are the species in the tree, and another one is another part are the branches, which have lengths. These branches are also called evolutionary distances. We will see how. So let's suppose we have a ancestral sequence here, and then as we go through time, those sequences diverge into different organisms. So we have sister species here, and then we see that in time step one, we have a transformation here to the left, no transformation to the right, then another transformation in the second time here, and then on the third time we have a transformation in the second species. So essentially what we see here is that when we go through time, we see an accumulation in number of transformations, what we might call evolutionary distance, which is which has a direct relationship with time. As we go through time, we accumulate transformations. Then we can use an evolutionary model that tells how likely transformations are, how frequent transformations are between the four bases in the DNA, and then use a bit of maths for arriving at this expression that tells the probability of transforming between the same state or different states as a function of time. And then from here, we can use a bit further math and arrive at this expression that relates the proportion of differences between two sequences and the evolutionary distance. And this is what we use in order to tell how far two sequences are. But here's the trick. Those distances are a conflation of rates, evolutionary rate on time. What does it mean? That at some point 0 in time, we want to go through a distance of 100 kilometers. And then if we go 100 kilometers per hour and we drive 1 hour, we will arrive at a distance of 100 kilometers. But then we can arrive at exactly the same distance by driving half as fast, but twice as much time, right? So we have essentially different ways of arriving at essentially the same distance. And this is what happens in these methods. We can't tell just by distance how much time has been through. So what the data are? The DNA sequences. So we have here different species of vertebrates and DNA sequences for all of them. And then we use the evolutionary model and computational tools in order to calculate the phylogeny, which depicts the evolutionary relationships between the species, but also the evolutionary distances, which are the branch lengths. So again, branch lengths are a conflation or a combination of evolution rate in time. How do we tell them apart? Here we have methods from divergent sum estimation, which essentially use the sequences and fossil information as data, and then three different parts of the model, which are called the substitution model, that tells us how fast or how things evolve into one another, and also the clock model and the tree model, and we integrate all of it over this using Bayesian statistics. In summary, how do we tell time and rate apart? We need to use this unrooted tree and explicitly model the rates, the branch rates, and tell information from time that normally comes from the fossil record. And that way we can have a time tree. What are the clock models? So the earliest mention of the molecular clock is due to Zuckerkandl and Pauling, that arrives at this relationship between the number of or the time between splits of different species and how many differences there were in globins, which are a very special type of protein. And essentially, what they say is, as we go back in time, we accumulate changes, but those accumulations are constant in time across the whole tree, which is called the straight clock. Then later on, different clocks have been proposed so that we can relax this strictness and propose, for instance, that different parts of the tree have different rates, or even each of the branches has their own rates. And then how do we tell information from time? From the fossil record, we can use node calibrations or tip calibrations, which are essentially using fossil information for specifying statistical distributions for the nodes, which function as priors in the case of node calibrations, or we can use explicitly the fossil specimens as part of the inference in the tree. And then they will be transmitting the time information through the along the tree as we carry out inference. A couple examples from my own research. So in this study, we used divergence time estimation and modeling explicitly the fossil record of different fish families in order to build a time tree that we later on used for biogeographic modeling in order to tackle the question about the relationship between the closure of the Isthmus of Panama and the evolution of this group of fishes, which is called snooks, the family Centropomidae, and the barracudas, which is the family Spherianidae. And we found an interesting signal of some probability of ancestral area reconstruction for which we don't have like really late separation between areas, which is sort of inconsistent with the idea that the isthmus closed around 2 or 3 million years ago, but rather that this process probably took a lot more time during the Miocene and probably part of the Oligocene. Here you can have access to the preprint, but this paper is about to be published in Journal of Biogeography. And then this is a study in which we used divergence time estimation to study this system in cultural evolution, which is how melodies from Gregorian chant evolved. And in this case, we were using the melodies themselves as sequences, and then the manuscripts as terminals or as species. And we found striking results. One of them is that we recovered two big groups that sort of correspond to East and West Frankish traditions, and they also recover a geographic structure in which we have the Rhine River in the middle of this part of the tree. We also were able to identify parts of the tree where we have very fast evolution and some parts that were inconsistent with historical information, for which we ended up discovering that part of, for instance, this terminal here actually was later in time. So we were informing history through phylogenetics, and also found that this has some monastic structure in the case of Cistercians, which were a monastic group, for which we find all of them forming a clade, which makes a lot of historical sense because Cistercians were a very close community that didn't quite mix with others, which is not the case of the Benedictines. which were scattered through the tree. I want to thank all of my institutions and funding agencies, in particular FAPESP, which funded my PhD, but also my current postdoc, and the BBSRC, who funded the project that hired me in Queen Mary, but also Stry and the Nat Geo Society that have funded my research during my the earliest part of my career when I was an intern at Smithsonian in Panama. And now I want to share a bit about Colombian culture with you. And I chose joropo, which is a way of music or a musical form from the Colombian and Venezuelan llanos. And it's essentially music for dancing, but it has other social contexts too. And it's essentially played with a cuatro, which is this small guitar, the harp, the bandola, which sometimes takes on the role of the harp, and the capachos, or maracas. And there is something interesting about the cuatro, and which is the golpes, which are essentially a combination of rhythm and sequence of chords, are the way different genres or subgroups of joropo came to be. And chords receive popular names instead of the standard chord notation, such as, for instance, puntito is what we call D major, or pata de perro is what we call A major. And playing in a particular tonality is implied by the chord in the popular name. So for instance, if we are playing something in D major, we are playing por puntito, or we might play agaban por pata de perro, which will be playing one of these golpe in A major. The setting is actually identical to the way people used to communicate music in the Baroque period for the Baroque guitar. For instance, we have this piece called Marionas por la E. E is the way of calling one of the chords, which we now might call with some standard name. And this was originally prevalent, and this is still prevalent, an oral transmission rather than academic, but there has been, of course, some evolution in this direction, and now joropo and especially the cuatro are taught in conservatories in an academic way. This is a beautiful example from Cholo Valderrama that you can listen later. And then this is what we call the llanos, which is essentially more or less consistent with the Orinoco Basin in Colombia and Venezuela, probably without these parts, but it's still questionable. And this is a very lively tradition that is still there today. So when I was back in 2017 doing field work in the llanos of Casanare in Colombia, I used to play in the haciendas where people allowed us to collect. And then we also named this species Ketostoma joropo back in 2016 as a way of raising awareness about this very important cultural form in Colombia. Thank you very much.